So we talked in our first lecture about the importance of the kidneys in regulating blood pressure and blood volume. This flow chart I want to refer to again just to summarize some of the concepts we talked about in the first lecture. So if we look at its role, the kidneys, in controlling blood pressure, this flow chart really summarizes both the intrinsic mechanisms, which means these are mechanisms that are acting right within the kidney, and the extrinsic mechanisms, which are those acting outside the kidney. So that can be via hormones acting systemically through the blood, or we can see here through the sympathetic nervous system acting through action potentials traveling to the renal nerves that act on the kidney to stimulate a response. So let's talk first about these intrinsic mechanisms again. We did discuss this already in our first lecture, but I just want to review that to make sure you understand. So if we have a falling blood pressure, that means we're going to have a decreased glomerular filtration rate. So the blood flow into the glomerulus will be decreased in through that afferent arteriole. As the cells that line the afferent arteriole, remember they have mechanoreceptors there. Those granular cells have mechanoreceptors that are going to detect a decreased stretch in the smooth muscle of that afferent arteriole, which is going to result in vasodilation. So as soon as there is less stretch in the afferent arteriole, that causes an immediate response of vasodilation. So vasodilation is going to increase the blood flow to the kidneys, and the goal of increasing the blood flow to the kidneys is going to maintain that glomerular filtration rate, because if we don't maintain the glomerular filtration rate, toxins are going to build up in the blood. We want to make sure we have good blood flow through that glomerulus, even in cases of low blood pressure, in order to keep the toxins and that electrolyte balance going throughout the blood with the help of the kidneys. So that's the goal of this intrinsic mechanism, that myogenic, which just means muscle producing mechanism. So the constriction and dilation of the afferent arteriole, we call that a myogenic autoregulation. It's regulating itself. So decreased stretch in the afferent arteriole causes a reflex vasodilation. Now, if we had increasing blood pressure, the opposite would be true. We'd see an increased stretch of that afferent arteriole, which would cause a reflexive vasoconstriction of the afferent arteriole to slow down the glomerular filtration rate. But in this case, we're looking at a falling blood pressure. And this falling blood pressure could be because of massive bleeding, for example. If someone is having a hemorrhage occurring, that's going to drop blood pressure very quickly, and the kidneys are going to respond reflexively using these intrinsic and extrinsic mechanisms to maintain homeostasis. So the intrinsic controls are to maintain homeostasis in relation to glomerular filtration rate. So these two mechanisms are just selfishly acting on the kidney itself. So falling blood pressure causes vasodilation of that afferent arteriole. The same thing occurs when we see a decreased flow due to low blood pressure passing through that distal convoluted tubule up here near the glomerulus, and as those macula densa cells that are in the walls of that tubule, as they detect a decreased flow of sodium, those chemoreceptors in the macula densa cells are going to secrete a chemical to cause, again, dilation of that afferent arteriole. And we talked in lecture last time that that chemical is nitric oxide, which causes a dilation of the afferent arteriole, again, acting to increase that glomerular filtration rate. The macula densa cells will also inhibit secretion of ATP in order to, again, relax that smooth muscle and dilate that afferent arteriole. So together, the granular cells and the macula densa cells are acting to increase the glomerular filtration rate, to increase blood flow in the kidneys when there's a falling systemic blood pressure. So those two are the intrinsic mechanisms. It's not as important that you know the names of these, the tubular glomerular mechanism or the myogenic mechanism. I'm not going to hold you accountable for those terms, but I do want you to know, understand that how the cells of the kidney, the granular cells and the macula densa cells, are working together to maintain that glomerular filtration rate in episodes of changing blood pressure. Now, if we look at the extrinsic controls, we can see here that the granular cells not only act locally on the afferent arteriole, but they also secrete a hormone to the blood, so they act systemically as well. So if we see a falling blood pressure, and again, those mechanoreceptors in the granular cells of the afferent arteriole are going to be stimulated to secrete renin with a decreased stretch of those afferent arterioles. That renin then is secreted to the blood, and it acts as an enzyme to convert another hormone in the blood that's not active until acted upon by renin. Renin will help convert angiotensinogen to angiotensin II. Then, as that angiotensin II flows into the systemic blood again and goes through the lungs, up in the lungs is an enzyme, which we said is that angiotensin converting enzyme. We just abbreviate it with the letters ACE, or we call it ACE. ACE converts angiotensin I, also found in the blood, to angiotensin II, and angiotensin II is the active hormone that causes vasoconstriction in the systemic arterioles. So now that's a little different than what we see in the afferent arteriole. The goal of the afferent arteriole is to vasodilate to keep that glomerular filtration rate going. But systemically, we're going to constrict the systemic arterioles in order to bring up pressure, because if we reduce the diameter of a vessel, that's going to increase the pressure in that vessel. That's going to speed the blood flow when we constrict it, and that's going to help to raise blood pressure so we can get enough pressure in those arterioles leading to the capillary beds everywhere else in the body, such as the brain and the heart and those vital organs. We're going to see increased blood flow, increased pressure to maintain the health of those tissues. Also, we see that angiotensin not only acts as a vasoconstrictor, it also stimulates aldosterone release from the adrenal cortex. This is the adrenal gland sits on top of the kidneys as part of the endocrine system, and it secretes aldosterone, which causes sodium reabsorption from the renal tubules in the nephron. And if we reabsorb sodium back to the blood, water follows that sodium, and that's going to increase the blood volume. So these two act together to increase blood pressure by constricting the systemic arterioles and increasing the blood volume. So again, this is a hormonal extrinsic mechanism, and its goal is to increase the systemic blood pressure. So the goal here is different than our intrinsic controls, which are acting only to maintain the glomerular filtration rate within the kidney itself. And then lastly, the other extrinsic control is through the sympathetic nervous system. So when we look at receptors for pressure in the systemic circulation, which we talked about in the cardiovascular system, remember the aortic arch, the atrial walls, and the carotid arteries have these baroreceptors for pressure, and when they detect decreased stretch, that's going to stimulate the sympathetic nervous system through the renal nerves to actively stimulate the granular cells of the afferent arteriole to secrete renin. So when we stimulate the sympathetic nervous system, that's going to stimulate renin production by direct activation, direct action potentials acting on the granular cells. So now this pulls us back to the nervous system. What type of receptors would be on the granular cells of the afferent arteriole to respond to sympathetic stimulation? Well, what we find are beta-1 adrenergic receptors that when stimulated, when they receive an action potential from a sympathetic nervous system nerve, the renal nerves, they are going to be activated as well, and they're going to stimulate renin in a response to that activation. So we've got hormones and nerves acting together to stimulate those kidneys to secrete renin and start that cascade of hormone production, angiotensinogen, angiotensin I, angiotensin II, and angiotensin II is going to it's going to stimulate vasoconstriction and that aldosterone secretion. We also see a little angiotensin II secretion from the kidney itself, so that kind of augments what's secreted systemically for that vasoconstriction. So there's a lot of information here, but it's really kind of nice because it's summarizing a lot of the information in a kind of a visual way. So I would definitely study this diagram and know the different portions of it very well, and it's going to really help you on our upcoming exam. So now we have to talk about tubular reabsorption in some more detail. So it's important to understand the importance of tubular reabsorption. We talked about last time, I showed you this little diagram here that really summarizes the three processes you need to know about. So glomerular filtration, we talked about, is that movement from the glomerulus, from the blood, to the Bowman's capsule. So that is really a passive process in the sense there's no active transport. We're simply squeezing the plasma out without any selective means. We're just squeezing it out based on pressure differences. So we lose our glucose, we lose amino acids, we lose important electrolytes, we lose some water in the process, and that needs to go back to the blood here through the peritubular capillaries and the vasa recta. So it's important we get that back to the blood, and that's what the process of tubular reabsorption is all about. It's getting that plasma that was squeezed out of the glomerulus and getting it back to the blood. Because our glomerular filtration rate is so efficient, remember it's 125 mils per minute, that we would essentially filter out all of our plasma volume in about 22 minutes. So that means in less than half an hour we would urinate away all of our plasma volume. So we need to reabsorb, we need processes throughout the nephron to pull those substances and that water back to the blood, and that's what tubular reabsorption is. Remember, we reabsorbed the water and the electrolytes in the digestive tract first, then it's in the blood, then we filtered it into the nephron. Now we need to reabsorb it back to the blood. So again, it's a selective process, this tubular reabsorption, and we want to make sure any important nutrients, organic nutrients such as amino acids and glucose, that those are totally reabsorbed back to the blood. We don't want to see those in the urine because we need amino acids and glucose for metabolism. And then the water and the ions are really regulated, which means we don't have to 100% reabsorb because it depends on our intake. Sometimes we're a little high on sodium, depending on our intake. Sometimes we might be a little low on sodium or potassium or hydrogen ion or bicarbonate ion. Those things are regulated with the help of hormones. So the process of moving these electrons and nutrients are both active and passive. And there's two routes that they can take. There's transcellular and paracellular. So if we look at this diagram, this is where the urine is inside the nephron. So we can assume that this is maybe the proximal convoluted tubule. So we have this filtered urine, we call it filtrate at this point. It's in the tubule, it's in the proximal convoluted tubule, and we need to get essential solutes and nutrients and water out of this tubule and back into the blood via the peritubular capillaries, because this is going to eventually lead to the renal veins and take it back to the general circulation. So we need to look at these processes then going from inside the nephron, across the walls of the nephron, into the interstitial fluid, across the walls of the peritubular capillary, and then it's back in the blood. So there's passive and active transport processes. If we look at the cuboidal cells up in the proximal convoluted tubule, we have two membranes that these substances have to pass. There's the apical membrane, which is here. So this is in contact with the filtrate, which is in the nephron. And then we have the basement or basolateral membrane here, which is the membrane before we hit the interstitial fluid and we're going back to the blood. So a couple of things that it's important to know. That when we look at the movement of electrons, or electrolytes, sodium is number one. So 80% of the ATP energy to move electrolytes in the proximal convoluted tubule is involved simply with sodium transport. So sodium transport is key here. And when we look at that movement, there are sodium-potassium pumps all along this basolateral membrane to actively transport sodium. So think of the processes we've talked before, and that is active transport via ATP using the sodium-potassium pump is occurring here at the basolateral membrane. So we're constantly pumping sodium out of the cell into the interstitial fluid. So what does that mean then for the concentration gradient inside the cell for sodium compared to inside the tubule of the proximal convoluted tubule? It's going to be less because we're constantly pumping sodium out. And then that's going to drive sodium in across the apical membrane. So again, sodium potassium pumps are all along this membrane here. And then what we find here are secondary active transport and facilitated diffusion occurring to get sodium in on this side. So all the activity, all the ATP is focused here to driving the sodium potassium pump via active transport, and then facilitated diffusion and secondary active transport is occurring at the apical membrane. So looking, let's see here, I'm going to quick pause. So if we look at the different types of movement here, we can see there's paracellular and transcellular pathways for movements of water and solute. Paracellular means between cells. So paracellular is between cells. And this involves leaky junctions between the cells. So there's simply spaces between these cells in which water and solutes can sneak through. So we do see some movement of water and solutes through the paracellular route. So there would be no energy required in this process because they're simply just leaking through. And we know that the proximal convoluted tubule is extra leaky, so that does allow those to follow their concentration gradients from the inside again of the proximal convoluted tubule into the interstitial fluid and into the blood. So we have to have a higher concentration here and a lesser concentration here in order for solutes to flow through the paracellular route. Again, through leaky, tight junctions is the paracellular route. The transcellular route is going through the cells. So here we can see the transcellular is going to require active transport, because if we have solutes such as sodium or any charged substance, they are not able to dissolve through that, again, that lipid membrane. So they're going to need active transport proteins to help get them through that cell. So what we find is we have facilitated diffusion, secondary active transport occurring on the apical membrane, and again for sodium we talked about the sodium potassium pump is on the basolateral membrane, actively transporting those solutes via the transcellular route. So again, it's really important that you know there has to be membrane transporters if they are not lipid soluble. Like again, the solutes that we see, the charged solutes, the ions that are important, and the amino acids, glucose, any of those charged molecules are going to need a transporter. So transcellular versus paracellular, it's important that you understand, again, sodium passes through primarily through transcellular. 80% of that ATP used for transporting solutes is used simply for sodium, because we find again that sodium is a major component of the extracellular fluid. So we pull that from the extracellular fluid that was filtered out from the plasma, and that's going to act as a concentration gradient, pulling other substances through the membrane as well, and we'll talk a little bit about that in a minute. So moving on then, we see that sodium is the most abundant cation in the filtrate. We have no trouble getting enough sodium in our diet, as we all know that. So again, it passes through primary active transport using that pump in the basolateral membrane, and then it passes by secondary active transport or facilitated diffusion through the luminal membrane or the apical membrane. That's what's right here. So luminal membrane, apical membrane, two different names, but again, where we see the inner lining of the proximal convoluted tubule is essentially what that is. And again, here's your basolateral membrane. Okay, so when we look at the energy, like I said, 80% of the ATP is used to move that sodium, and that provides the energy by creating concentration gradients for reabsorbing other substances. So when you talk about secondary active transport, that's where sodium is expending the energy, but it's bringing another electrolyte along with it, or it might be bringing glucose along with it. So either way, we're using the energy for sodium to pull other substances out of the nephron and getting it back to the blood. So organic nutrients, and when we refer to organic nutrients, we're talking about amino acids and glucose. Those are all reabsorbed by secondary active transport with the help of sodium. So there becomes a point, though, that there's only a maximum amount of carriers in the membrane of the nephron to transport substances. So if you think of these carriers that are present here on this luminal or apical membrane, there's only a certain number of protein carriers here to take and pull substances out of the urine. If those carriers are all full, because there's a lot of, say, sugar in the plasma that was filtered into the nephron because of someone who has diabetes, for example, these transporters are going to be maximized, and they can't pull all of the glucose out of the urine and put it back into the blood. So we call that the transport maximum for that substance. So when the carriers are saturated, which means they're full because they're transporting as many as they possibly can, the excess of that substance is going to be excreted in the urine. Again, in the case of a person with diabetes who has really high blood sugar because of a lack of insulin, all that sugar is filtered out into the nephron, and the nephron simply can't keep up in transporting that glucose back to the blood because those carriers are full. So water is reabsorbed by osmosis. Again, there's this concentration difference that's been created by the movement of sodium, and we know that water follows solute. So if sodium's being reabsorbed, water's going to follow, as long as there's holes in that proximal convoluted tubule for the water to pass through, because we know that water can't pass through a fatty plasma membrane. So we have these special holes that are called aquaporins. We find those in the proximal convoluted tubule. We also will see that they are hormonally controlled in the collecting duct, but that's how water flows. So water flows by osmosis. Now remember, that's not an ATP driven process. So water flows passively as long as there is a concentration difference based on the sodium movement, which is an active movement. So that movement of water across the membrane is simply by osmosis through these holes in the plasma membranes of the proximal convoluted tubule, which we call aquaporins. Remember, anything that is fat soluble that can dissolve right through the membrane will follow through by diffusion. So this is just kind of a visual showing you all the different types of transporters we see in the luminal membrane here. So we see we've got these secondary active transport. Sodium is using the ATP to make a concentration difference, and glucose, amino acids, ions, vitamins, just they're along for the ride. Water, here's passing right through via an aquaporin. Notice that this is blue, so this is passive transport. As long as we have sodium increasing in the interstitial fluid, that's pulling water. It's creating an osmotic gradient, sucking water into this interstitial fluid and then into the peritubular capillary. And then again, some of these other ions are just leaking through by the paracellular route based on concentration differences. And if we're constantly losing sodium here, that causes a positive charge to build up here in the interstitial fluid, which is again going to leave it more negative inside the cell, and that's going to pull some other substances in based on their concentration gradients. So this movement of electrons changes the charge, making it more negative inside the cell of the proximal convoluted tubule. So again, we can see these transport proteins, if they become saturated, for example, this one that's moving glucose, if all these along the proximal convoluted tubule up here are saturated with so much glucose, they can't simply transport anymore, and any excess glucose passing by here remains in the nephron and ends up in the urine and causes a positive test, and that's going to tell the healthcare worker that there's a condition of maybe diabetes occurring if there's excess glucose over a long period of time. So when we look at the proximal convoluted tubule, it's the site of the greatest amount of reabsorption. Most of the sodium and the water that's been filtered out from the glomerulus is reabsorbed right back to the blood at the proximal convoluted tubule. So 65% of the sodium and the water. All of our nutrients, which means glucose and amino acids, all of those, 100%, are reabsorbed back in the proximal convoluted tubule. So we should not see a large amount of amino acids any amino acids. Some small amounts of proteins can leak into the urine, but very little, only a trace amount we should have seen in the urinalysis, and no glucose should be present in the urine because it's all been reabsorbed. And then we see a lot of ions, other ions like magnesium and phosphate and chloride and potassium, and then small proteins are reabsorbed in that proximal convoluted tubule. And then from there, the filtrate enters into the loop of Henle, and this is where we can see some selective movement of water and salt, particularly sodium, potassium, and chloride. So we'll begin in the next video talking about the loop of Henle and how we can get a concentrated urine by controlling the amount of sodium and water reabsorption.